my name is Jacob Bernard, and like my previous topic, I'm going to be talking about bioinformatics. However, today I'm going to go into a little bit more specifics of bioinformatics software and programs that are used within the field. So to start off with a little bit of an overview, we're going to start off with a bioinformatics review, kind of touch on some things that we talked about before, such as sequencing, to make sure that we're still up to speed, understand where we are, and understand what's being talked about. Next, we'll go into some software tools and major programs that are used within the field that people will be using to create their projects and study various things. Next comes the databases. These are databases that have been created and kept up over the years to have a large amount of data to be accessed and compared against when doing studies. Next comes our software languages. So once we have these databases and these software tools, we need a easier and more efficient way to be able to work with these tools and these projects. So we use things like Python and Perl and Ruby and Java, and we create packages for them that are like other packages that we've used before, and they are implemented to create a much more ease of use for the programmer. Then that will lead us into some more specifics and going into the details of kind of how these languages work and how one would get set up and what it kind of looks like, and that specific would be BioPython. And finally, a little bit of a conclusion. So to start us off, we'll go into what is bioinformatics again. Bioinformatics is the application of informatics techniques to obtain, store, and interpret large quantities of biological data. Computers are used to collect, store, and manage the data from research so that it is available to other researchers as the basis for further study and discovery. So to touch on a topic that we talked about a little bit last time, sequencing. Sequencing is important so that scientists understand a subject's genome in order to get a view into the organism's physical makeup and physiological abilities. So they will sequence a sample of the DNA to extract these base sequences such as the A, G, C, and T codons within our DNA. Now, once we have this sequenced DNA, we need to be able to do something with it. This is where these software tools come into play. There are two major categories when dealing with software tools and programs in the field of bioinformatics. The first category is primer creation. So a primer is a short single strand of RNA or DNA that serves as a starting point for DNA synthesis and is generally required for DNA replication. So some programs that would be under this category would be things such as AutoPrime, MethPrimer, PrimerQuest, and other such programs. The next category is bioinformatic work programs. These are general programs that will do things such as search, analyze, categorize, and such on. The programs that will do things like this would be BLAST, BioEdit, ORF Finder, and things of the like. This leads us into our next category of databases, which is a little bit next further. So once we have kind of our sequenced DNA and these program tools, we need another set of data to compare it against. This is where these databases that have been created and kept over the years come into play, so that we have a kind of control group to compare and do studies and projects with. So to start off, I'm going to discuss a little bit of these databases and kind of what they are. So the first one is the EST clusters database. This is a repository of assembled transcript sequences from express sequence tags in mRNA. So this is kind of like patterns that will be seen in order to discover new genes from already existing data. The next is the immigrant genes database. This database contains genes that have immigrated or been transferred from one species to another. So what this would be used for is to track certain genes that over the years have mutated or evolved to jump from one variable species to the next. The next one that I'm going to talk about is the leukemia database of genes. This was developed to help biological and medical science communities to easily access all information on the genes that are involved within leukemia disease. The next p53 tumor protein gene is very similar, keeps track of all of the p53 tumor genes for further research and study. The pancreatic cancer gene database, once again, is very similar and provides information on the genes that are involved in pancreatic cancer. And this data is targeted to help the biological and medical sciences community for easier access of the latest information on genes causing pancreatic cancer. The next is the TB drug target database. This database contains information on the anti-tuberculosis drugs and the target proteins for the treatment of tuberculosis as a disease. And finally, that brings us to the acronyms databases. This is a database that will analyze large medical texts and records to go through and search for acronyms, so that you can simply go through and search an acronym from a large text and find it very quickly instead of having to thumb through a large book. Now, finally, we have kind of all of our tools that we need for the process. So we have access to a database, we have a DNA sequence, we have programs, and we have programs. And now we need a streamlined and efficient way to manipulate and work with this data. So this is where these projects were started in order to kind of make an ease of use way of using this data to similar program styles such as Python, Java, Perl, and Ruby. So during the Genome Project in the 90s, multiple projects were taken up with similar goals to streamline code packages for major languages to be used. These packages would have versatile, reusable modules and classes to use in various bioinformatics projects. So these projects then became BioPython, BioJava, BioPerl, and BioRuby. These were created to make it much easier for people to kind of transition and be able to use these packages in a much easier way instead of learning a whole new language. And finally, this leads us down to our last little deep path that we're getting to here, which is an actual bioinformatics kind of language package itself known as BioPython. So we're going to kind of go over a few little things of like the history of this and how it came about and what it's used for and kind of what you can expect from a system like this. So for a first little brief history of creation and functionality, BioPython development began in 1999 and it was first released in July of 2000. The initial scope of BioPython involved accessing, indexing, and processing biological sequence files. While this is still a major focus, over the following years, added modules have extended its functionality to cover additional areas of biology. So this was kind of started along and piggybacking on with the Genome Project in order to make it easier for scientists and computer scientists to kind of come together and work easier and more efficiently to create projects. Now for a little bit of a general design description. Wherever possible, BioPython follows the conventions used by normal Python programming language to make it easier for users familiar with the Python language already. So for example, there's something called the sequent object in BioPython, which is considered one of the central objects. Now this object can be manipulated in a manner similar to Python's string and list objects. So if you know how to already use some of, say, Python's major data structures and functionalities already, you're going to have a very much easy time transitioning to using the BioPython packages. It is also designed to be functionally similar to other bio projects such as BioPerl, BioRuby, and BioJava. This, once again, is for ease of use and familiarity between languages, not only between creating a package similar to a language, but creating a similarity between all languages. BioPython is able to read and write most common file formats for each of its functional areas, and its license is permissive and compatible with most other software licenses, which allow BioPython to be used in a variety of software projects across various aspects of bioinformatics. BioPython can be used for all sorts of functionality, from parsing, connecting databases, sequence slicing, transcribing, and nucleotide complements. So, BioPython is a set of libraries to provide the ability to do things of interest to biologists working on the computer. Its job is to make your job easier as a programmer by supplying reusable libraries that you can focus on answering your specific question of interest, instead of focusing on the internals of parsing a particular file format. So just like you'd use, say, a CSV parser within your normal Python packages that would be included for free or something that you could import or download, BioPython is very similar. You would import this package, utilize some of the functionalities within it that already happen to be extremely similar to Python, which makes it much easier to use, connect it up, and it works pretty easily. And as you can see from this picture right here, it looks very, very similar to working with normal Python code and working with normal Python lists. In this, sequences are literally just lists, just as in Python is. So you can work with these, you can get the index of this sequence, you can transcribe this sequence, do all sorts of endless possibilities with it. So, in conclusion, bioinformatics is a vast field that has lots of uses, and today I'd really liked to focus on the software aspects of it and really focus in and show you what the world of computer science has done for this and how it can be utilized within this field. And now we have things like packages and languages that are exactly similar to Python, Java, Ruby, and Perl, and we have endless possibilities to go on. So thank you very much, and here are my sources.